This lecture talks about bacterial genetics. A patient comes into the clinic with a vaginal infection. There's a thick greenish-yellow discharge, and the patient is complaining of a burning sensation during urination, severe itching, and a swollen vulva. The presence of gonorrhea is confirmed within 6 hours via a nucleic acid amplification test, or NAT. The patient is started on an antibiotic treatment immediately. The patient wonders, how did the results come back so quickly? How do you know that it is for sure gonorrhea? In this lecture, we will explore the structure of DNA and learn about how DNA makes each microorganism unique. So first, an introduction to the bacterial genome. A genome is the complete collection of genetic material in an organism. The bacterial genome is 0.4 to 9.4 million nucleotides depending on the species, but the human genome is 4 billion nucleotides. Now for an exam, I'm not going to make you memorize any of these details. You do need to know the definition of a genome, but other than that, this is just kind of interesting information. So a human genome has 1,000 times larger than E. coli, but only 6 times more genes. So 24,000 genes in a human versus 4,000 genes in E. coli. 85% of E. coli codes for protein, or E. coli's genetic material, but only 2% of our genome codes for protein. So what is the rest of it doing? We're going to learn a little bit about that as we go through this lecture. So a chromosome. A chromosome is an individual unit of DNA. Chromosomes are usually condensed, so they contain chromatin, which is wound around histones, or DNA on histone proteins. Now bacteria don't have true histones, but they have histone-like proteins. If you look at this picture on the previous slide, you can see in this lower right corner here, this is the bacterial chromosome. Bacteria typically have one chromosome, and it's circular, although there are some exceptions to this rule. Plasmids. So plasmids are small circular pieces of DNA that provide extra genes. These extra genes are not essential for the life of the organism, but they do usually provide some type of benefit, like the ability to utilize certain nutrients or the ability to survive in the presence of an antibiotic. Going back to this picture again, these small circular pieces of DNA are plasmids. A gene is a segment of DNA that codes for a protein. Phenotype versus genotype. Phenotype is the physical characteristic that is expressed by the microorganism, while genotype would be the genes that are present in the genome of the microorganism. So if I can give you an example, let's talk about a resistant type of bacteria. Let's say malaria, the Plasmodium parasite, has become resistant to pyrimethamine, which is one of the drugs that's used to treat malaria. So there's functional resistance, which means that we can see that if we put pyrimethamine on top of the malaria parasite, it doesn't kill it. So that would be a phenotypic expression. We could physically see that the parasite's still alive even in the presence of the drug. And then there's the genotypic analysis. Does it have the gene that makes it resistant? The gene would code for a protein that alters the structure of the binding site of the antibiotic, or and somehow makes that antibiotic get pumped out of the organism or destroys the antibiotic. So the genotype would be, is the gene present? And the phenotype would be, is the gene actually expressed physically? A lot of malaria parasites have genes for resistance, but when you actually put them in the presence of the drug still works. So just because you have a gene doesn't mean that it's always physically expressed. Now we're going to talk a little bit about the structure of DNA. So Watson and Crick kind of took the credit for discovering the structure of DNA. It was actually Rosalind Franklin's X-ray crystallography of the structure of DNA that led them to the final structure. So she did major work on this project, got absolutely no recognition, and then she ended up dying from exposure to X-rays. So this is always my shout out to Rosalind Franklin, who was the true discoverer of DNA. But back then, they were men and she was a woman, and women in science were never regarded as anything, so these guys took the credit for it. So on that happy note, we're going to go through an overview of the structure of DNA. So when we look at the structure of DNA, DNA is double-stranded, and if you know what DNA stands for, it's deoxyribonucleic acid. It is double-stranded. It is an alpha helix. And the strands run in opposite directions, so it's known as being anti-parallel. And again, that means that the strands run in opposite directions. So the major building block of DNA is going to be a nucleic acid, or a nucleotide. And a nucleotide has three major parts: deoxyribose sugar, a phosphate, and a nitrogenous base. And there's four bases: adenine, or A, guanine, or G, cytosine, or C, and thymine, or T. According to Chargaff's rules, A is going to form bonds with T, and these are two hydrogen bonds. While C forms three hydrogen bonds to G. Adenine and guanine are known as the purines. Cytosine and thymine are known as the pyrimidines. One way that you can remember that is the word pyrimidine has the letter Y in it, so does cytosine, so does thymine. Now uracil for RNA is also a pyrimidine, and that one doesn't have the letter Y in it, but that one you're just going to have to remember. Now the difference between purines and pyrimidines is the number of rings. While purines are going to have a double ring structure, pyrimidines have a single ring structure. So if we were to draw out the basic structure of DNA, we have alternating sugar and phosphate groups, and they're anti-parallel. So if those are the sugars, then we have the phosphates. And that's what makes up the backbone of DNA. And in the center is where we're going to have our bases. So if we have a T on this side, we have an A on this side, and it's held together by two hydrogen bonds. If we have a G on this side, we have to have a C on this side, and that's held together by three hydrogen bonds. And if we have an A on this side, we will have to have a T on this side, which is held together by two hydrogen bonds. These strands have a 5' end and a 3' end. And because the strands run in opposite directions, the 3' end of this strand is up here, while the 5' is down here. Hopefully all of this was a review from previous classes. If not, you should review the textbook a little bit more about DNA structure. So now we're going to talk about some of the differences between DNA and RNA. First of all, the name. DNA stands for deoxyribonucleic acid. While RNA stands for ribonucleic acid. And that's because of the type of sugar. So we have deoxyribose sugar, while RNA has ribose sugar. The bases are a little bit different. So in DNA we have A, C, G, and T. But in RNA we have A, C, G, and U for uracil. DNA is typically double-stranded, while RNA is typically single-stranded. Double-stranded DNA is the typical formation or conformation of DNA. But we do have single-stranded DNA viruses. So viruses can take on a weird shape to their DNA. Likewise, single-stranded RNA is the typical conformation, but viruses can exist as double-stranded RNA. And there's a lot of examples of double-stranded RNAs like microRNAs and siRNAs and all sorts of stuff, but we'll kind of leave that discussion for now since this isn't necessarily a genetics class. The location of the DNA is either in the nucleus or the nucleoid. The nucleoid would be in bacterial cells because remember, bacteria don't have a nucleus. Otherwise, in eukaryotic cells, it would be in the nucleus. Now, RNA can be found all over the cell, so multiple locations. RNA is going to start out in the nucleus or nucleoid, and then once it's replicated, it can make its way over to the ribosome. The ribosome itself is made up of RNA, so it can be floating all the way around the cell. Although RNA is a lot more unstable than DNA is because of its single-stranded nature. Now, the function of DNA, it stores the genetic code. And RNA helps make proteins. The major types of DNA are really just genomic. And then if we are a eukaryotic cell, mitochondria have their own DNA. The types of RNA include tRNA for transfer RNA, mRNA for messenger RNA, and rRNA for ribosomal RNA. So ribosomal RNA makes up the ribosome, tRNA is used during translation, and mRNA is the copy of the DNA that's made during transcription, which is brought over the ribosome during translation.